So today I'm going to cover about comparative genomics. So as the word implies that comparative genomics is analyzing multiple genomes and to make sense out of the genome. So suppose we have human genome now and we have several uh, mouse genomes, rat genomes. So you want to make sense of the data. That is comparative genomics by combining different. It's more into evolution, but there can be several other things as well. So we'll discuss comparative genomics, structure sequence again, and today I will discuss drug discovery using marine genomics, where you'll use extensive uh, comparative genomics, and then we'll have some training session where you can train yourself and prepare yourself for the. Uh, so now when you're doing any comparative genomics, you have to ask yourself, what am I doing? Question you have to ask. Is why I'm doing, is it doing, I'm doing for studying evolution or doing functional annotation, the purpose. And then you can define your purpose, and then you have to chalk out your raw materials from which you want to solve. Suppose you want to study a fungi, then fungi only matters. Human or plant leke to kuch hoga nahi. You need to have lot of samples of fungi. We will cover some of those areas. And then comes genome assembly. What is genome assembly? You have small pieces in a DNA fragment. When you are doing assembly, you will assemble them into a page like this, a full genome. That is in genome assembly, and something like I showed you in a bookish way. And then you do gene prediction. So let's see what is gene prediction is. So DNA has a lot of information. So a gene is a piece of large entire chromosome. So in a chromosome, how many places different genes are there? So as you see, the word is broken into small pieces, with colorful pieces. This colorful unit will make gene. And we'll see some example. And functional annotation. Functional annotation is identifying meaning. Using, say, if it is English literature, you will look into dictionary. In the genomic sense, we'll look into databases. And to make a overall meaning of what you are getting in the genome. And comparative genomics, what is comparative genomics? Now again, I'm asking you and you are asking me. So it's like comparing two genomes or more than two genomes for the purpose of understanding what is similar, what is dissimilar. And then annotation is a difficult task, and it takes lot of gravity to do that. The reasons you will understand why, because you are doing for 20,000 or more genes at the same time. So what is the ultimate goal of doing annotation or comparative genomics? We all want to know how gene and protein works and how that implies to function of the living organism or function of the protein or in the cell. Or so Bruce Albert has defined. So ultimate hallmark is functional implication of the gene, protein, RNA, whatever you are looking at. And if function can be defined in different ways. So function can be defined as a network to the cellular biologist, and how network is working. It could be metabolic process, like how Krebs cycle is working after one reaction, what reaction is happening. And in the cellular compartment, people are looking into, okay, this protein I have, it is expressing. So where it is going? It is working in nucleus. It is working in, say, it is working in nucleus. It is working in cytoplasm. It is working in endoplasmic reticulum, Golgi. Where it is working? All this you want to understand, and there a lot of that can be a hint you want to get it about function from the genomic study and comparative genomic study. So functional annotations can be biochemical function, biological function, regulation, interaction, expression, all sort of things, and which you understand from basic biology will implies together. And then what need to be annotated? Protein need to be annotated. Protein domain, signal peptide, transmembrane domain is there or not there, or how many proteins are kinases out of all proteins of an organism or all gene of the organism. How many are coding and how many non-coding RNAs are there? If it is a prokaryotic operon is existing or not, a cluster, a critical functional cluster on the chromosome exists or not. So these all you need to annotate. And what is protein domain? A domain is discrete structural element that will fold independently. So yesterday I gave you example of serpin. Serpin is a single fold. So fold, and it has its own function. It can have on the average in a multi-domain protein, it may have 20 to 100 amino acid long unit, but it can go up to 400, 500 amino acid as well. And motif is small stretch. So we'll come back to serpin, and serpin has reactive center loop, which is a motif, and entire serpin is a domain. Okay. So that part will come when you again cover serpin again today a little bit. And then in the coding region, what you are looking for? You are looking for enzymatic coding, receptor, toxin, kinase. You have to define. Suppose you are studying plant, you have a different definition, because you are maybe looking at how I can improve the quality of wheat. So you are looking for certain traits of wheat. And if you are looking at rice, you are looking for certain different traits of the wheat. So that is, when you look into literature, the traits are already available. So science is always done on the previous knowledge. Like coronavirus do not come out of certain. There was a cause previously a coronavirus or Merck coronavirus, and from there you get it. So basically we had some knowledge already. So that's why one can get vaccine very quickly. Otherwise, for a totally new organism or new system, it was difficult. So basically, you invite previous knowledge and improve the previous knowledge, or apply the previous knowledge. Then you can use these non-coding riboswitches, RNAs, microRNAs, CRISPR, and then pathway prediction and all sort of regulatory elements, promoter, enhancers, and that in cancer we will see even in detail about this. Then when you I just briefly explained what is genome assembly. Let me show you some of the genome assembly and genomics technology. So you all are hearing about genome assembly. Suppose human genome is known, and our coronavirus 2 genome is known now. When you're mapping after sequencing the small reads to the known genome of known organism, then it is called mapping-based genome assembly. And when you don't have the so there is the blue known genome is not known, and from that you tell the program, okay, I have reads, I want a solution, and then it will make a de novo assembly. And it's like this. Suppose you first person who made the car, he really had an idea of the car. Maybe he took it from Balgadi or horse rider. From there, he took the original idea. Rather than using animal, let's use 4 wheels. Same applies when you do not have the same genome. You have some idea, longer stretch of DNA will be made. It may not be initially very correct, but then if you do not have, you better to have something. And so that's why it's when you make a initial genome assembly, draft genome assembly, because there is a scope for improvement. So even after 20 years of human genomics, there is a good scope that human genome will still be improved in the next 20 years. So this much, but on the draft, one can start working and look for better one time to time. So this is 2 type of genome assemblies. And when you assemble genome, what you see is, if you can visualize an assembly, you can do assembly with different software. enormous software. Some of them, the easiest one I have noted here: CLC Bio, Mira, Ray, Meta, Velvet. You can use once you do it. What you see here is that, let me have the browser highlighter. So you have okay, I have taken highlighter. So this is assembled genome, and then these are the small individual reads. Okay, so this is from an actual genome sequencing project I have taken for you to have a look. And then you will ask, okay, how did you draw this? This is not a drawing. This is just a visualization from a software called software is Tablet, and Tablet is available from if you look genome assembly and Tablet, you'll find in Tablet you can see the how genome assembly look like. Okay, so you see that individual reads are there, and one reads is different than many of the other reads, and one reads are small pieces, right? So you have this reads are coming here, and then from here onwards, one reads is continuing forward. There is a gap. Maybe later on there is one starting somewhere here, and so on, and then another one is starting. So so the next generation sequencing is called massively parallel sequencing, because this is taking care of that no stretch is left, and then the multiple many millions of the small reads are available, and these reads are indeed causing you a look like this. And from here you can call mutation, because now the C is all the time C. In one case here, you can see that A is there most of the time and C is one time. So this can be error. But suppose this is constantly happening. C's, you are getting C, you are getting the C all the time. Maybe out of 30% time C you are seeing, then it is a mutation. Yeah. So depending on how, what kind of data you have, you can pick from I also some mutations, and otherwise you will apply some software when you are doing picking up the mutations. So there are a lot of genomic terms. Reads are a small piece of DNA which is chopped on, and you get it from sequencing read. On the sequencing read, initial assembly is done on contig, and contigs can be continuous. So or this can be discontinuous. So therefore, one contig is only one continuous stretch of DNA, and second one is the first one and second one. There is some space. I can't see the marker myself, but let me. So so you see a gap, and 2 and 3 has a bigger gap. So gap will remain there. So contig will merge into scaffold, and a scaffold will be merged into the chromosome. So what you expect? Contig to be bigger or chromosome to be bigger? Definitely chromosome will be bigger, because more than one contig will form into scaffold, more than one scaffold will form a chromosome. So chromosome is the largest unit. And many times reaching to chromosome is not possible, because you do not have the assembly required data for assembly. Laser pointer I have taken. So you have the gap here, which is retained here. If it is found here also, you will see those gaps. And so if you are doing analyzing the data in ensemble, you can see those gaps in human genome or other genomes as well. So let me tell you what is N50. You may have known many times that if you are in genomics, N50 is you place your contig scaffold or chromosome from largest one, any unit to take from largest to smallest, and then start summing the length of contig 1, 2, 3, 4, and if it is more or equal to the 50% at the fourth contig, the size of that contig is called N50. So total nucleotide 50, and there is a definition 75. 75 means it will be, if 50 is fourth, 75 will be somewhere third or second one. And this is one of the one of the criteria to tell people that how good or bad our genome is. If a genome is assembled to lesser contigs, N50 is reached in less and less contig or less and less scaffold, your genome will be of better quality, because you have lesser pieces remain. And so in this case, say I have taken 40 MB genome, so 20 MB is reached in fourth contig, and that's why. So therefore, it's written the higher the N50, the better the assembly. So this is kind of one of the threshold used in genomics to see how good your assembly is. And maybe you need to improve it. You can't just make one and say this is my draft assembly. You will need to draw five, like any drawing. Suppose you're drawing, you draw many figures before you come up and say this is my final figure. So given given that we go to next level. So here in one of our papers, we have used genome assembly reporting, and this is reporting from Roche 454, Illumina, Iron Torrent, and then we made a hybrid of all. So we have taken Roche. So genome assembly was 32.2, 32.5, 32.6, 32.2 MB in hybrid also. So the number of contigs here, Roche has a larger scaffold, larger contigs. So the number of contigs are less in Roche. In Illumina is length, since the read length was smaller, the number of contigs are more, and torrent even worse. But in hybrid, you see that 661 total contigs. What does it imply? That in this case, contig N50 is 114. In this one, you see 165. In this one, this 5,000 something. And then in the hybrid one, which is final, a mixture of all, we have this 79 sequences were there in and 50 of 121. So this is the highest we could achieve. And then obviously we have made different. So the final one we have taken into the consideration. And if same thing, if you look at the N50 for a scaffold, again, this is 116, 67, and this one is again better. So ultimately, which one we will choose? We'll choose this one. Or one prior to this, that can be this one. So that's how you decide how you want to take which genome you have taken, and then you are going to review. A reviewer has to agree that what you have performed is correct. Otherwise, they will ask one to do it. And then in this was a fungi. The genome size is genome size is 32 MB in approximation. It varies in a little bit, but it's approximately 32.2 MB and 16,298 genes. Once you have assembly of genome, you want to predict the gene. So basically, genome is full of ATGC, and in ATGC there is you'll see a pattern of a start of UTR regions, exons, and exons first coding ATG, and then stop codon, and you see a kind of a signal where the gene is starting. And in the genome, when you are doing gene prediction, you will software will know that look for UTR like region, look for up next, look ATG, then continuously go until here. GT is the intron which is cutting out splice donor and acceptor. Look splice donor and acceptor and look for exon, intron until you reach a stop codon. And then you look for UTR region, and then once pass through, go for next non-coding region, and then non-coding region. Then start looking for UTR like region, and then these signals. So based on that, algorithm can figure it out where are the genes in the genome. So this is what is genome gene prediction. And so how one is doing genome prediction? Suppose you have a context, you make a gene prediction using one software, two software, three software, and merge it. And one of the easiest software or very popular software is Augustus, and which is easy. So it's easy to go. You can load in any of your DNA, and you can actually get the gene out of it. So it's online also available. You can run online. These you cannot run online. So some of them, if you are interested, you can go to Augustus, give us a piece of DNA and run. You'll see a gene prediction result of that. Okay, so then comes to genome annotation. When you're doing genome annotation, what you're doing is that you have your genome. Now you are using a lot of BLAST and other algorithm. And you are, suppose I'm studying fungi, I will use fungal genome. BLAST has NT database, NR database, UniProt database. Look for homology detection. Look for protein domains. Maker will pick. Maker will also do the analysis. It's a similar way. Maker is a pipeline, and then you will also do with a specialized tool called BLAST to go and pick number of genes which are getting annotated. So in this, I'm showing you that we have pre-marine fungus genome, and we could annotate approximately 60% of it, 8 or 70% of it, but red ones are not annotated. So there are a lot of whatever annotation strategy you will use, there will be a lot of areas where genome will be remain unannotated, and that you have to take care. And we have to minimize it. But it's common that about 20 to 40% of genes will be remain unannotated, matching to the unannotated genomes of the previously known genomes. So you will get a lot of unannotated material, and that's minimizing that is a major challenge. So when you're saying annotation, what type of genes are there in the genome? A gene which is known gene, like suppose the gene is predicted matches the entire length to a known gene, then it is called gene of that name. So like for example, BAM46 is gene, and which has known function, so it's called, it can be taken exactly. Putative ones are ones where the gene contains region that are conserved in the gene, but not fully, so you refer them as BAM40 like or similar. Similar, for example, unknown genes are not genes whose function is not known, matching to genes where the function is not known. So if the previous gene is unknown, this gene will be unknown. And then there will be genes which will be remain hypothetical because they do not contain any signal similarity. Maybe you have made a computational prediction, over prediction, and you're getting a gene which is not matching anywhere in your database, or there can be a database limitation also, but those genes are called hypothetical. So these bottom two will create problems. So if we are getting 30 to 40% unknown or unannotated, these database has 20 to 30% or 20 to 40% of the unknown genes already. So you will get hit to them, and then the annotation will be not perfect. When you're doing annotation, you will also look. So there are 16,000 genes. Now in 16,000 gene, if you want to analyze, it's a very tough task, right? So what you do is that you look for gene annotation, gene ontology actually, and gene ontology basically classifies. This is a good graph here, but they classifies it into three categories. What is the molecular function of the gene or protein? Which cellular compartment it will go and stay, or will stick. So it will take it from the and then which biological process it is. And this is from our own paper, and there are different ones. So in a graph, when you see there are numbers which are in many fold. But suppose you are looking on RNA binding, suppose one of this class, then what it has done, that what this analysis has done, that you only have to look 100 genes. If you are looking for one RNA binding or 5 RNA binding protein, Now you do not have to look 16,000. You just have to take one of the bins, one of these 600 genes. And then you have those 100, and that's the advantage of using gene and ontology analysis. And that's good to have actually. So in our study, one of our study, we identified, we did gene annotation, and from annotation we could see that our species were primarily belonging to these fungi mostly, and these are all member of a fungi class called Sartoriomyces. And so that's a good example actually. And after annotation, we could recognize that these, suppose we don't know the name even, we can actually pick it. In this case, we were knowing the name, so it was easy for us to guess. But from genomics also, everything is going correct, then it is falling to same class. So as I told you that we'll look for domain. So what you do is that you will look for protein domain and look for the different kind of, in these three species, we have identified different domains. And you see that we have identified P54. And their number is much more in the one species, Pestarotiopsis, while lowest in the Populoropsis. So what is this showing, that if you are only looking for P50 proteins in these species, you can only tap on these. So I have just made a graph for you, but the data is also there in the. We will have the data also in the when you are publishing or when we are keeping with us. Yeah, so one can look into that class of protein with carrying these domains. Or if you're looking for a zinc finger CH2H2 transcription factor, you will look only for these. That creates protein domain analysis, will help you in that aspect. And then when you're reporting the genome analysis, you are also so in this case, you are also comparing with other species what is their genome size and what is their genome numbers, gene numbers in the per species. So green one is the one which we have covered, and then we see the genome size is 32 MB, but number of genes are much higher higher than one from Nectaria, which has a 51 MB genome, but the number of genes in this case is higher. So this is crossing 1600, as you saw there, and 16,000 actually, and this is lower than 16,000. So 15,000 something. So one can compute a comparative analysis you see in multiple species done to give a hint how the gene distribution of our genome size is going on. And again, coming back to comparative genomics, you need these databases again and again and again. And we will see today in comparative genomic sense, these databases. Yesterday we saw in a phylogenetic and gene detection sense. And obviously this, again, I'm repeating it, but these are from chromosome to genomic DNA, from mRNA to protein, and you have different databases. And we have seen some of them, and some new ones will be covered today. So again, I'm coming back to Terpenins, and when I told you the Terpen is a single domain, that means the one domain is carrying, one single domain is carrying core Terpen, but Terpen can differentiate between each other by having some extra regions in the core. So let's compare a human body. Human look different because of some hair on the chin or on the head, or some female will have longer hair. So but the core structure is core structure of the body is head to toe, and these are extra beautification. So protein domain is same, like the core hard mass, which we call it in Hindi. That skull body is the same of human. Similarly, proteins will have same domain most of the time, and then extra bulges or extra region will make them specialized. And on the protein, there is a one domain, and domain will have a motif. So in this case, a reactive center loop is the motif. And we saw yesterday, yeah, I think we have it here. Okay, so this motif looks like this. So 20 amino acid, yeah, this is 5 here and 15 here. So 20 amino acids, and this have conservation and all, and these are, so that is motif when we are looking into previous definition. So, and we already saw that how this is working in a branched pathway. It will control proteases by sacrificing itself. And obviously, we have seen a lot of functional implication yesterday. What I'm going to discuss about this V6 protein and their comparative genomics of V6. I just deliberately skipped yesterday, and so that we have it. Today's class will cover. So HSP47 is collagen chaperone, and if you want to cover the chaperone, we have to collagen chaperone. We have to cover these species, as I told you. Then this is comparative genomics again coming in, because you are comparing more than one genome. So genomic data has to be available. And at that point of time, this many data was available. Today, when we started the analysis, today this is more than 100 species available for us to study. And then databases we have covered. And today what I'm going to, that one point I'm going to cover. Another point is going to cover is that V1 serpin. So you see that fishes have one copy of this, and this is duplicating. So we'll go and see in database this expansion also. So, and yeah, so we're coming back to HSP47. What we identified in this, that Japanese lamprey has a locus, which you see that the yellow one is the HSP47 with respect to human. And this gene is also carrying P2RY6, and P2RY6 is conserved in this locus, and also it can go to the other fishes also. You've identified that this is conserved, where you will get some more genes, and this is the first cluster. And in fishes, this cluster is maintained. And then this is orthologous to each other, because from human to fishes, lamprey, this is a single copy on the same locus. So therefore, it's called ortholog. From lamprey to human, all other species, one copy each. But fishes have the second copy of HSP47, which is a yellow mark yellow, and then one copy in all with respect to human is ortholog for all. But the second copy in fishes is paralog, originating by duplication event in the fishes. So and you see that this is also conserved. So this gene and this gene and the third gene are called paralog in the fishes. If you're taking Amazon molly or Medaka, in there they are Amazon molly is there, or let's see, Taki fugu is there, Taki fugu is there, Taki fugu is not there. So wherever the species has the 3 copies, those have these are paralogs of each other, because within a species. And so the definition of paralog is that paralogs are originated by whole genome duplication event within the fishes, or in this case it is fishes, but in general by duplication they originate within a single species. And orthologs are genes which are occurring by the event of speciation, means one species will have one, because if mouse has one and mouse convert into rat or whatever rodent it is, they will also have one copy because of gene pool transfer from one species to from their ancestor. That is ortholog. So this one I'm only saying that this one is a lamprey, which is in primitive fish, has in fishes one copy, and also in tetrapod one copy. So this is ortholog in all species, because it's probably because of a speciation it has originated. While these are from this gene duplication, 2 more copies are originated. So these are called paralogs. So when we look, we saw that the genomic mapping, we did genomic mapping in the databases and we identified this. Then when we look, plot this on a evolutionary time scale, human has one copy, all the tetrapods have one copy. And then what we see here is that the lampreys also have one copy, not the second copy. And this gene is not found in invertebrates. So 0 copy. Okay. So, but this gene has 3 copies, 3 copies, 3 copies, 2 copies, sometimes 1 copy, 2 copy, 3 copy, and also some examples are there where 4 copies are there. So, so what does it implies? It implies that this an evolutionary scale, if you see before this line, all have 1 copy. So that means only in this sub branch there is a multiplication of HSP47 or collagen specific chaperone, and then this is due to whole genome duplication. But why they need it actually? So what is the physiological implication? So when we looked into the collagen genes also in these species, the corresponding number is doubled compared to what is found in tetrapods and primitive fishes. So that could explain that then it require multiple chaperones are there. So chaperone for activity of chaperone proper folding of collagen, you need multiple collagens are there. So basically you need corresponding chaperone also. So by comparative genomics, one can predict. And then you need to further do the experiments. We have not done the experiments, but we hope that others will follow up. Yeah, so that is one part, and then one more part which I am going to cover related to portion of it was covered in the theoretical portion. So we look for drug discovery using marine genomics, or you can do marine or other organisms. So we looked at marine fungus, and marine fungus are, as you see, they're very colorful, and they pose a lot of secondary metabolites. Secondary metabolites are future drug-like compounds, which have potentially very good pharmaceutical, antibacterial, or anti-cancerous drug properties. So there are several examples of it. Cephalosporins are found in this species. Antibiotics, anti-cancerous, Aspergillus luteus has this compound. Ascopolaride is found in Ascopolopsis, which we have covered extensively. And then normally in fungi, there are PKS, NRPS, and terpene, like 3 type of drug producing compounds, and these compounds are then coming into these compounds are then coming into the forming a 4th kind of hybrid with by joining the it can be mixture of this and this, or mixture of this and this, or mixture of this and this. So the 4th compound is hybrid. So there are only 4 potential compounds in living entities or in fungi, and these are actually normally found in the cluster. So there's one gene, and then you need a accessory like transcription factor and other components. Entire cluster is required for functioning of the core gene. This is core gene, and for production of the drug. So what will happen that this transcription factor and other genes will help production of protein from this gene, and this gene will form a larger protein, and from protein one amino acid will be produced, and then it will fold properly to form a structure, and we will see how genomics help there. So it's not that marine fungus were not studied recently. They were studied, but since on our land itself there are so many diversification is available that people focus on the land because of penicillium, discovery of penicillium from penicillium has dramatically shifted our research to look for a future drug-like compound from fungi on land, and also people slowly were focusing on marine sources. You see from 1960s onwards, very much, but with the advent of genomic sequencing, this has gone very fast, and that portion I have contributed to the yellow ones in an extensively. And so how it works actually? So it works like this: you collect the sample from marine source, and then you do collect bring marine sample to lab, and in a lab you try to grow them, and if it is growing, you can actually take them for DNA extraction, do whole genome sequencing. Initially, you will do whole genome sequencing using Illumina 454 and iron torrent, and then you will do the genome sequencing. After genome sequencing, you will do genome assembly, and as I told you, genome assembly can be de novo and or mapping when the known marine fungus or known species is known. Genome is known, you will do it in version 1, and then you will do hybridization of genomes assembly using 2 or more genomes, and then this is called first version of the genome assembly. And then if you have money and if you have a clear agenda, you can actually also do whole genome sequencing with third generation of the DNA sequencing. Third generation is the long read PEG-BIOS solid or nanopore, and then you will take help of hybrid assembly using a set of the softwares, and these softwares are available to us. So so that you can do the error correction. Doctor. So the read assembly, and then you can do long read assembly, and then you can hybridize with the previously known assembly and make a better assembly. And why it is required, I'll explain you. It is required in some cases, in some cases you don't need. So therefore I'm calling V1. If the version is 1 is okay, you can go ahead. If you need that one, you can, and you have one needs also money, right? So everything boils down to ultimately to money. So we have done assembly of 2 kinds. Then we can detect repeats. What is repeat detection? Repeat detection will find whether there are transposable elements, repetitive elements which cause problem in the gene prediction, and further analysis will detect and mask them. So we'll just block those areas wherever we find repeats. And then we do gene prediction using Augustus 3 or Maker pipeline. And then we do genome annotation, which I have already explained. And these are the tools: BLAST, BLAST2Go, HMM, PFAM, Interpro, KZ. There are several ones. Then we detect the biosynthetic gene cluster where the drug-like compounds are present using set of tools. So initially, you can take help of this general annotation by using BLAST and HMM, and then you will do the specialized analysis with NTS-MAS, Cluster Finder, EvoMine, and then you will also able to pick the drug-like compounds from chemical structure from here, and also you start grouping biosynthetic gene clusters into the families, do some network or analysis, and then you will get kind of a set of future drug-like compound. And yeah, so this is how the entire pipeline is working. So in one of the case, we have identified the from marine derived Scoporopsis, we have identified this anti-cancerous compound, and which we went and characterized fully. So what happens in this one? There is a PKS2 laying in this somewhere in the genome, and then this one will produce this red portion, and then PKS is required for production of that final one, which will help in circularization, or the first one, whichever way you want to call. So this is the first one actually. So, but when we are doing assembly, we are not getting the full scaffold. We are getting the broken one as you see. And that's why you need third generation of the sequencing to come in and help. In this case, we were able to solve it. We had to do assembly 142 times and do annotation. Keep on checking. We kept on checking. It took us almost 2.5 years, that we were sure that if it is a drug, this has to be present nearby on the. So basically, these are looking two independent clusters, as you see, but actually they are hybrid genes are actually on a same cluster. So one PKS plus one NRPS is required for production of this drug. So finally, after several years of computational analysis, we could gather this 2.5 years to be precise. We gathered this. We knew everything. This gene, this everything we're knowing. Only this portion has to be how you'll fuse. And we do not have third generation facility. So, but by doing assembly, changing parameters, doing with different algorithms, we could achieve. And then you see that this is located on scaffold 447 in the final one. And that's when it helps. So basically, where it comes from. So it came from a comparative analysis that similar compounds are in other fungi are produced by this kind of a hybrid cluster. So we get it an idea that if this drug is produced by this organism, this will be the final one will be the one single cluster rather than two clusters. So and then we did RNA-seq experiments for this, and what we see that there are. So as I told you that different clusters are required for production, and now we know that these are the cluster members of the clusters which are all required. A transcription factor is required, and which is zinc finger here, transporter is required. All other components are required. So in different condition, how they are expressing. So if you want to have a production of these two in a, so these two in has to be in a coordinated way. So you see that not much difference in these two expression of these two. And then you need, if you need a higher, if it is a drug you want to produce in a higher quantity, modify biotechnologically, you need to derive mutants, you need to derive, you may need to change. So now you know the cluster, you can change the transcription factor, swap the transcription factor, and get, so basically you can produce more amount of drug, which is produced by this cluster. And that's we did by mutagenetic analysis, by mutating the cluster, and also you can do it by genetic engineering. So that's why the larger clusters are important, and larger third generation of sequencing in this case is important if you are looking for a drug-like compounds from the genome. And while doing the analysis in different studies, we identified that terpenes can be only of two classes. By comparative studies, we have identified that terpenins can be terpene cyclases or terpene sequelases, and not yeah. So either these are carrying these domains or carrying these two domains and grouping into two different type of terpenes produced by different fungi. By just using domain analysis in several fungi and their terpenes, we could identify. In the drug-like compounds will be producing clusters will be a lot in the fungi, but this is RNA-seq expression. So in laboratory conditions, they are not required because these are secondary metabolites. In a normal condition and happy hours, they don't need fungi don't need it. When you give them some stress, maybe high salt concentration, low, maybe heat shock, maybe other kind of shock, you give them a lot of sun, then they produce. They have this organism, Calcareosporium, has 50 of them. So maybe 5 of them is expressed in one condition, 5 of them are expressed in other condition, and 5 of them are expressed in another condition. So basically, condition optimization is required to get this 50, and most of them are anti-cancers or antibiotics properties. So basically, if you want to explore further into genomics, has provided that they are total number 50. If you want to explore, and we also did for some want to explore further, one need to optimize extensively in laboratory under which condition which one is produced. And you see that they have similar domain architectures. You will expect a very similar compound maybe, but with a few changes, there are new antibiotics. So, but there are 50 in the genome. So that shows that there is a massive potential. And what we did was so as I told you that we needed to see biotic and abiotic stress. So we send our staff to, in one of the case, to marine Baltic Sea in Kiel. And then fungi we already have, bacteria we already have. Then we started challenging bacteria with fungi. But before that, we wanted to have a composition of bacteria, which we get it. So in this, we did genomics, and we found the Bacillus and Vibrio strains are very common in water in this region. And then we started challenging our bacteria, and we see different expression profile of the these clusters. And I'm not going into much detail of that because there are a lot of biotechnology involved there, and a lot of other people have participated in the characterization. So what we did was we combined several fungi and biosynthetic gene clusters, and we could identify overall, say, 10 organisms, 311 biosynthetic gene clusters. And these biosynthetic gene clusters are, as you see, are 310, means at least 310 compounds are produced from 10 organisms, so 10 odd organisms. So basically, you have 30 per organism on the average, if I count, and that's a very good amount because the drugs can be even modulated with the condition. So at least 300 drugs are drug-like compounds are potential, and this has a very massive potential. And 10 such drugs we have already characterized. And while doing that, we recently published 2021 in Marine Drug. We did a comparative genomics of marine Rhodotorula genome with several Rhodotorula genome known in NCBI. And what we identified, the genome size is 19 to 20 MB in most of the cases. Highest is 22 here. And 20 MB genomes, they produce very similar biosynthetic gene cluster 19. And in one of the case, they have 21, and one exception is that they have 5. So we made prediction ourselves, and then we saw that one NRPS, there are 1 to 4 terpenes are there. One other kind of unknown kind of cluster is there. Several of this putative clusters are there, and one of the cluster is fatty acid cluster found in most of the conserved in most of the species. And then one of them where the low number we have identified, that one has none, but most of them have one. And we were looking for a drug by metabolomic studies. We can identify this is coming from a fatty acid, and then we went on and tracked. So normally you will expect this to be present in a cluster. We identified this cluster, and then did the phylogenetic analysis, domain prediction, characterization. But for this cluster to be active, in literature, it was known that these kind of species or many fungi also need an exoinolylase enzyme, and then we have characterized that enzyme separately, so that if it is cluster is there and this is not found in the organism, organism is not capable of producing. But metabolomic study has already proven that the compound was produced. So therefore, in genomics also, these has to be present. So, but we have to show them. So that's why we carried out the analysis in the West. And so this is one alone cluster will not work. You need a cluster, and then one more gene which is away from the cluster. And one protein is required for proper production. And that shows that when you are doing comparative genomics and genome mining, it will help your process of drug drug discovery, and you will get a hint where we can focus. Otherwise, if you have 50 or 70 clusters, you can't just start tapping one by one, or it will take many years. Okay, now those portions which we have done. Now let me tell you what we are going to train today. So we will look for one gene, maybe SERPINA1 or any other gene. But today we look it from not from gene perspective, from the cluster perspective. Then we'll also do it in ensemble, same thing. Then we'll look for the cluster. Let's see if the ensemble is working today. And then we will also use UCSC Genome Browser, which is the third browser, so that you will be able to generate a cluster. We'll use SERPINA1 or one of the cluster and in 2 species, so that we can see how it is working. And then if you want to have a drug like compound prediction, this tool is there, and we can at least go and see how load the example data or example output and see how it's looking. While doing will be not possible because it takes several days to produce the result, but we can actually play with it. And then I again thank you for your time, and we will have a practice session now.